My screen? Yeah. Hello, everybody. Welcome. This will be the final session, final lecture for this particular class, for this particular topic that we have been looking at, that is basics of neurobiology, right? So we had started discussion about the sensory modalities in the previous lecture. Um, just a moment. Just a moment. Yes. So as I was saying, we have already begun with the discussion about sensory modalities in the brain in the previous lecture itself. There are two major sensory modalities that we have already covered. These two are the auditory and the visual modalities, right? So how are we seeing things? What is the, rationale behind how we are perceiving the world around us, right? What is the pathway that that information is carried to the brain? And what are the cells responsible for carrying that information? Similarly, we have also seen the similar kind of outline for the olfactory we'll do today for the auditory system or auditory sensory modality also, right? So these were two major ones that we had covered in the previous session. Now that we know, we have a sense of what is a sense what is a sensation versus what is perception, which is also what we have covered in the previous session. Now we can finish the three sensory modalities that are left really quickly, right? So one of them is touch, that is the sense of touch. The second is the olfaction, and the third is gustatory senses, that is taste, basically, right? So these are the three that we are going to cover today. Let's just begin with that. So first that we'll be talking about or discussing is the touch, right? So what is touch? Touch is basically defined as a direct contact between two physical bodies. In neuroscience, when we are talking about touch, it refers to special sense by which contact with the body is perceived consciously, right? So the sensation is what? It is a physical contact with something. And what is touch to us? It is perception of that physical contact, right? So that is what is touch with respect to brain involvement, with respect to what we are feeling, right? So this touch can be active as when you move your hand or some other part of your body against another surface, or passive as when someone or something else touches you, right? For instance, you have a friend nearby and the friend starts calling you. So he'll pat on your shoulder, for instance, that is a passive touch, right? So somebody else is initiating that touch, right? Or for instance, there was a snowball that was thrown at you, right? So the snowball came and hit you. So that is also, you will feel it through a touch sensation, but it was a passive touch, right? You did not initiate that to touch that snowball. It just came and it hit you, right? Active touch could be like, for instance, I am moving this mouse so that my cursor will move, right? So that is active touch. I know that I'm doing this, right? If I'm writing with a pen, that is also an active touch, right? If I am washing my hands, that is again active touch, right? So passive touch is used for naming objects or describing sensations. Active touch is used when hand manipulates objects, right? So washing hands, my hands will go in a certain direction because I want to clean the surface of my skin that is present on the hands, right? Over here, I'm using the mouse cursor. That is why I'm actively rotating the mouse so that my cursor will also rotate, right? Uh, then uh, Yeah, active and passive modes of tactile stimulation excite the same population of receptors in the skin and evoke similar response in the afferent fibers, right? So, are the receptors different for when we are actively touching something or when there is something else that is coming and bracing against our skin or touching us, right? No, the population of those two neurons are actually the same, right? How does it differ though, right? But my body knows, or I know that I did not do this. I was not touching that snowball, it just came and hit me, right? I know the difference between the two, right? How? Because they somewhat differ in cognitive features that reflect attention and behavioral goals during the period of stimulation, right? So when active touch is that physical activity is still going on, at that time the cognitive area of your brain, which is has something to do with motor coordination or attention to something, because you are trying to write in a particular kind of language, so that is active, right? So the cognitive part of it is what will be different, right? If you talk about your hand, which has the maximum, touch response. So, skin and touch receptors are present throughout your body, but they are most abundantly present in the hand, right? And especially in the fingers, because you do a lot of work through this, right? And you most of the things that you feel the sensation of, right? You will probably do it via your hands and your fingers, right? That is why hand has a maximum number of touch receptors. And these touch receptors we had studied earlier that they are mechanoreceptors, right? Once we were going through that entire chart in the previous session, we had seen that touch receptors are mechanoreceptors, right? So they are mechanically activated, basically, not chemically, not electrically, but mechanically, right? So the tactile sensations in the human hand arise from four different kinds of mechanoreceptors that are present. The names of these four receptors are given over here: Meissner corpuscles, Merkel cells, Pacinian corpuscles, and Ruffini endings. Right? The sense of touch can be understood as a combined result of information provided by these four systems acting in concert, right? So it is not like only the Merkel cells are going and recognizing something, or only Ruffini endings are going and recognizing something. It is usually a combination of these mechanoreceptors, that when this combination will get activated, that is when the sensation of or perception of touch will occur, right? So this is a representation of these. You can see over here is marked the Meissner corpuscle. This is the Merkel disc receptor. Then this is Ruffini ending, and this is where is the Pacinian corpuscle, right? Over here in front of these, you can see that there are two these different abbreviations that are written. RA1 in front of Meissner corpuscle, RA2 in front of Pacinian corpuscle. Merkel disc receptor has SA1 written over here, and Ruffini ending has SA2 written over here, right? So what does that mean? We'll study in a while, right? So it has something to do with whether it has a rapid action. RA is rapid action and SA is slow action, or it has a slow action, right? So we'll see that in the coming slides. So each receptor responds in a distinctive manner depending on its morphology, innervation pattern, and depth in the skin, right? So it does have some kind of a unique ability to respond, right? And these receptors are innervated by either slow adapting or rapidly adapting axons, right? So this is uh, adapting actually, right? Rapidly adapting or slow adapting, right? So slow adapting fibers or SA fibers respond to steady skin indentation with a sustained discharge, whereas rapid acting fibers stop firing as soon as the indentation is stationary, right? Sustained mechanical sensations from the hand must accordingly arise from the SA fibers, right? So, for instance, there is something that is going to keep pressing on your hand, right? For instance, if I was wearing this watch, right? So the watch is going to keep having that uh, sensation on my hand, right? So probably because it is a sustained sensation, it will go via this slow adapting fibers, right? Rapidly adapting fibers will not even respond to the sensation because it is not a momentary sensation. It is going to be there on my hand for the entire day, maybe at night also, right? So that is where the difference arises over here. The sensation of motion across the skin is signaled by this RA fibers, right? The type 1 fibers terminate in clusters of small receptors in the superficial layer. So this is the superficial layer, epidermis, then dermis. So type 1 fibers are basically their endings are reaching over here, right? At the margin between dermis and epidermis. Whereas type 2 fibers innervate the skin sparsely and terminate in single large receptors in the deeper layers of the dermis or in the subcutaneous tissue, right? So that is where these type 2 fibers are going. The RA1 fibers, right? That was the Meissner corpuscle, are the most numerous tactile afferents in the hand, right? So Meissner corpuscles are the most numerous kind of mechanoreceptors that are present in our hands. They reach a density of approximately 2 per millimeter square at the fingertip in both man and monkey, right? Yeah. So over here, this particular table represents the receptor, the location where this receptor is most abundant. What is the kind of axon diameter that the neurons over here have? What is the kind of stimuli they respond to? Then what is the frequency range that they respond to, right? And etc. Right? So you can go through this. Over here, you can see the receptor fields in human hand are the smallest at fingertip, right? So we have studied earlier what is receptor field. It is basically a measure of of like whether something can be perceived or how like close by something is that we can still distinguish that these are two objects, right? Over here, it could be that could be what is receptor field. For instance, if again, because this is the simplest thing that is always in my hand, so that is why I always take the example of this mouse, right? So if you can see over here, the mouse has this ridge that is present, this ridge that is present, and then the rest of it is plain, right? So when I'm feeling it with my fingers, I can make out that, okay, this is a different region, this is a different region, this is a different region, right? But if I began feeling with my hand, right, I will probably not be able to make out the differences that well, right? As well as I am able to recognize the differences from my hand, I might not be able to recognize it that well with my palm, right? So that is what they are saying that over here in the hands, these dots represent, kinds of receptor fields, right? So the receptive field sensitivity, look at how good it is, right? But over here, these bigger portions are actually the receptive fields, right? So the distinguishing ability is lesser in the palm region of the hand than the fingertips, right? Similar for the entire body. The entire body, other parts of the body have even lesser receptive field, right? They also have mechanoreceptors because they also have touch sensation, but the receptive field is even lesser, right? Then the tactile information is processed in the central touch system. So this is the system that is present in the brain. It comprises the dorsal column tracts of the spinal cord, relay nuclei in the brainstem and the thalamus, and a hierarchy of regions in the cerebral cortex, right? So, this tactile information enters the cerebral cortex through the primary somatosensory cortex in the post-central gyrus of the parietal lobe, right? So, this is the somatosensory cortex. We have studied the somatosensory and motor cortex lying next to each other when we were looking at the cross organization of the brain, right? We have gone through all this central sulcus, lateral sulcus, all these major structures we have already studied, right? So, over here, you can see this is the area which has this a region where this tactile information is actually processed, right? It is represented over here as this 1, 2, 3, and 3B, right? So the neurons in the SI are at least 3 synapses beyond the receptors in the skin, right? What does that mean? That the neurons that are present in the skin, the ones that are receiving signals in the skin, the axons of those neurons that are present over here, they are not directly forming having cell bodies in the central touch system or anything, right? They are relaying information to the central touch system, right? Which is at least 3 synapses away, meaning that one neuron is connecting and relaying information to the second neuron, that is one synapse. Then second is connecting and relaying information to the third synapse, second neuron, right? The second synapse. And then third neuron is conveying information to fourth neuron. That is the third synapse, right? So at least 3 synapses are beyond they are, right? So it could be more than 3 also. Their inputs represent information processed in the dorsal column nuclei, the thalamus and the cortex itself, right? So, each cortical neuron receives input arising from receptors in specific area of the skin, and these inputs together are its receptor field. So, over here you can see that there is some color coding that is done over here in this areas of primary somatic sensory cortex, right? And what does this color coding represent? That is given over here, right? So, the muscle splinters will be recognized over here, right? SA1, RA1 receptors will be recognized over here. Complex receptors and proprioception will be recognized over here, right? Active touch over here, and so on, right? So, there are specific areas in the cerebral cortex which will receive information from specific areas on the skin, right? And these are the various kinds of receptive fields that are shown over here on this in this example, right? Also, we perceive that a particular location on the skin is touched because a specific population of neurons in the brain is activated, right? So, what is the use of them being different? Like areas differing according to which specific area of the skin is being activated. Why is that? Because whatever area of the brain now is activated, we will know that, okay, that brain area is connected to this area in the fingertip or this area in the hand. So we know the sensation is happening in this fingertip or in this area in the hand, right? This experience can be experimentally induced by electrical stimulation of same cortical neurons, right? What does that mean? That even if we were not feeling anything over here, right? Even if in our brain, that particular cerebral cortical area was stimulated, right? Through an electrode or something. Then, for instance, somebody stimulated this 5 number area, right? So I will feel like I am writing, although my hand will be at rest, although I will not have any pen or pencil around, but I will still feel as if I am writing, right? Or feel as if I am, washing my hands. Some kind of active touch response. I will feel like I am doing, although I am not doing it, right? So the primary somatic sensory cortex comprises four cytoarchitectural areas, right? So these are the areas we have seen before also. These are not areas. They are 3A, 3B, 1 and 2, right? These areas are extensively interconnected such that processing of sensory information at this level involves both serial as well as parallel processing, right? So there is some information which will be relayed across these areas also. It will first go to 3A, then to 3B, then to 1, then to 2. And some will have a parallel processing, such as some active responses also happening. Meanwhile, I am facing some passive touch response also, right? So one brain area is contemplating this, and the other is contemplating the other one. So parallel processing is also occurring. The neurons in the 3A and 3B receive inputs from lateral and medial zones of the ventral posterior nucleus of thalamus and project their axons to areas 1 and 2, right? So this is how actually they are interconnected. So they are receiving information from thalamus and they are projecting information to area 1 and area 2, right? You can see it via these arrows that are marked, right? So this is serial processing and this is like they are directly jumping to 1 and 2. Areas 3B and 1 receive information from receptors in skin, whereas 3 and 2 receive proprioceptive information from receptors in the muscles, joints, and skin, right? So in the previous class, we have already seen what is proprioception. Over here, we know that proprioception is also under the cognition of this touch control center in the brain, right? The central touch system in the brain. So the somatosensory information is conveyed in parallel from 4 areas of SI to higher centers in the cortex, right? Including the somatosensory cortex, secondary somatosensory cortex, that is S2, the posterior parietal cortex, right? This is secondary somatosensory cortex, posterior parietal cortex, primary motor cortex, and posterior parietal cortex, right? Then over here also there is an columnar organization of the somatosensory cortex, as you can see over here, right? Columnar organization meaning that the neurons that are actually coming, earlier we had said, right, they are at least 3 synapses away. So this is one neuron, it is relaying information to the second one, which is relaying information to this third one, which is finally taking it over here to this area, right? Then we come to the fourth and fifth, that is smell and taste, right? Why are we discussing or beginning the discussion of both the smell and taste together? We will understand, right? Because the kind of things that they perceive are similar, right? Both of them have chemical modalities that are perceived by them. So through the sense of smell and taste, we are able to perceive a staggering number and variety of chemicals in the external world, right? These chemical senses inform us about the availability of foods and the potential pleasure or danger, right? So smell and taste also initiate physiological changes required for digestion and utilization of food, right? Once you study the digestive system and how hormonal changes work, right, you will see that there are certain enzymes that are secreted only in response to us knowing that we are going to have food, or us smelling some food, right? In many animals, olfactory system also serves as important social function by detecting pheromones that excite, that elicit innate behavior or physiological responses, right? Pheromones are like volatile compounds that it has been shown in insects and in other invertebrates, especially that they are useful for contacting or communicating between one another. There is also some kind of a deliberation that pheromones might be present in higher organisms and even in humans also. And these pheromones in higher organisms, they basically help as a secondary sexual feature only, because they help kind of how to put it, I'm not sure. In basically the attraction between opposite sexes, that is what its function is deliberated to be, right, in higher organisms. So odorants volatile are basically volatile chemicals that are perceived as odors and are detected by olfactory sensory neurons in the nose, right? So these chemicals that we perceive are or odors that we perceive are volatile chemicals which are known as odorants, right? Then there are a large number of olfactory receptor proteins which initiate the sense of smells. We will look at them very briefly. The sensory neurons that are embedded in a specialized olfactory epithelium that lines part of the nasal cavity, right? So this you can see is the nose, right? We are smelling something over here. It is going upwards. It is going to this olfactory epithelium, right? So, olfactory epithelium is very closely linked to the olfactory bulb that is present in the brain, right? Olfactory bulb is actually a part of the brain. That is why it is said that nasal epithelium or olfactory epithelium is almost at the junction of the brain, right? It can that is why some infections they can directly infect the brain or affect the brain through this region also if there is some kind of a loss of barrier over here. So the sensory neurons are embedded in a specialized olfactory epithelium that lines part of the nasal cavity over here, approximately 5 centimeter square in area in humans, and are interspersed with glia-like supporting cells, right? So over here you can see these are the olfactory cells, olfactory sensory neurons, and these are the supporting cells, and these are the basal cells also that are present, right? The olfactory sensory neuron is a bipolar nerve cell, right? You can see that it is bipolar. There are two extensions that are coming out from the cell body, meaning it is bipolar. The single dendrite extends from apical end to the epithelial surface where it gives rise to numerous thin cilia. This is a dendrite again, right? It is giving rise to numerous thin cilia that protrude into the mucus that coats the nasal cavity, right? These cilia basically have receptors that recognize odorants as well as transduction machinery, which is needed to amplify the sensory signals and transform them into electrical signals in the neuron's axon, which projects from the basal potential or which projects from this basal pole of the neuron to the brain, right? So this is the pathway that it basically takes. These are the cilia, these are the odorant molecules. In blue, the cilia has olfactory receptors. These receptors are going to take up or respond to these molecules, right? And then these neurons will take that information of what kind of receptors are responding to what kind of molecules. That information will be carried across this cribriform plate, which is like a part of the skull, right, to the olfactory bulb, which is part of the brain, right? In this olfactory bulb, there will be other neurons that will further relay this information to the olfactory cortex that is present particularly in the brain, right? The axons of olfactory sensory neuron pass through this cribriform plate, which is a perforated region in the skull above the nasal cavity, right? The axons then synapse in the olfactory bulb. You are seeing that what is a synapse? It is passing information to another kind of neurons, right? So that information transfer is happening through the small gap, which would have chemicals and neurotransmitters, which is known as a synapse, right? Which is the first relay in the olfactory pathway, right? So this is the first synapse that is coming in place in this olfactory pathway. Humans have approximately 350 different odorant receptors, whereas mice have approximately 1,000, right? So the organisms such as mice and some other rodents, etc., who live in the wild and who have basically developed around this concept that the sense of smell is more crucial to them because they are also nocturnal animals, right? Because they are nocturnal, anyway, not much light is there. So their eyes are also developed, but they are not as developed as our eyes, for instance. But definitely their nose or their olfactory sense is developed much more than ours, right? That is how they survive. So that is why they have around 1,000 receptors, while we have only around 350. Although odorant receptors belong to the G protein coupled receptor superfamily, they share sequence motifs not seen in other superfamily members, right? So they are also GPCRs. They are also kind of G protein coupled receptor. The short form is GPCR. So they are also a part of this very large receptor family, which is GPCR. It is also a very, um, important or crucial receptor family because most of the drugs that are targeted are to these particular receptors, right? But despite them being a part of this GPCR, they have some crucial characteristics that are not similar to the other family members of this GPCR proteins, right? GPCR receptors. Significantly, the odorant receptors vary considerably in the amino acid sequence, right? So that sequence uh, difference is definitely there. A second smaller family of chemosensory receptors is also expressed in the olfactory epithelium. This is the olfactory epithelium. What is another family that is expressing over here? It is basically known as trace amine associated receptors or TARS, right? They are also G protein coupled, but their protein sequence is unrelated to that of odorant receptors, right? Again, over here, where is the difference that is arising? It is again in the amino acid sequence, right? That is completely for the stars, that is trace amino acid receptors. It is completely different from that of the normal odorant receptors. These stars are encoded by a small family of genes present in humans, mice, and fish, right? So it is not really present in all animals also. So this will basically help you understand how basically even though we have only 350 receptors, still we will be able to recognize like thousands of smells, right? How does that happen? Because these receptors work or they get activated in different combinations for them to respond to different kinds of smell, right? So that is what is given. Different combinations of receptors encode different odorants, right? For instance, these are examples of odorants that are given, right? This first odorant will stimulate only this receptor, right? Sorry, what will be a result of its stimulation? That the odor that we will get will be rancid, sour, or goat-like, right? But if along with this receptor, this particular odorant was also able to stimulate this green receptor and this orange receptor, then the smell becomes like violet, sweet, and woody, right? If, for instance, instead of this second odorant molecule was present and this purple one was not activated, only the green and orange one were activated, then the odor is like sweet, herbal, and woody, right? So what does this suggest? That they're different, like the smell will depend upon what is the combination of odor receptors that the particular odorant molecule can actually activate, right? Now we can assume that these odorants are not present, right? But we have the odor receptors present, right? We have access to these odor receptors. If we can even artificially stimulate only this blue one or this blue purple, uh, this light blue and this yellow combination, then we will receive a sweet orange and rose smell, even though this odorant is not present, right? Even if we are artificially able to stimulate this, it will always feel like we are smelling something sweet, something like an orange, or some kind of a rosy smell we are getting, right? So that is the perception that our mind will get, right? Because our mind doesn't know what is there, right? It is deciding based on what is a receptor sequence or what is a receptor combination that is getting activated. If that gets messed up, if too many receptors are activated, then the receptors might be saturated and the brain might be not be able to distinguish the smell, right? Also, these odorants are encoded by nose in the nose by dispersed neurons, right? Also, to sum this slide up, this is how, even though we have around 350 odor receptors, still thousands of smells we can, identify, right? Even we can identify a difference between something as simple as a lentil or a dal that we have, almost every day. If it is, tasting a little sour, like something like sambar, or it is tasting normal, or it is very salty tasting, or if somebody has put a lot of chili and that volatile nature of that chili is coming to your nose, you feel that this smells a little spicy, right? This smells very plain, something like that. You can figure out even in the same smell, you can distinguish the intensity of the smell based on which odor receptors are getting activated, right? Then the different types of odorant receptors are expressed in several core zones of the olfactory epithelium of the mouth, right? So over here, this is an example. If we are talking these K20, 21, L45, A16 are different classes of odor receptors, right? Over here, this is marking A16. So you can see they are present at multiple places in this olfactory epithelium. L45 is also present in multiple places in olfactory epithelium. K21 again, multiple places it is present. K20 again, multiple places. And mostly you will see that more or less it is bilaterally symmetrical. The regions it is present in this hemisphere it is also present in this side of the nasal epithelium, right? Similarly over here, if the expression is over here, it is more or less similar expression over here also, right? And this final one is going to mark all odor receptors, and you can see that actually the entire nasal epithelium has odor receptors that are very specifically lined, right? But when we look at a class of odor receptor, you will see that they are widely spread, right? So each receptor type is expressed in approximately 5,000 neurons that are confined to one particular zone. These neurons with same receptors are randomly scattered within the zone so that neurons with different receptors are interspersed, right? Why is it scattered? Because even if, for instance, this region was somehow damaged, or you had cold and this region was covered with mucus, still this region can smell something, right? So, or for instance, if the smell is so faint that it cannot even reach this particular region, over here it can be sensed, right? That is why it is so scattered. Although one zone may have more receptors for a particular odorant compared to other zone, all zones contain a variety of receptors, right? For instance, this particular zone, if we look at, contains A16 also, it contains L45 also, and somewhat contains K20 also, right? But so it has a combination of different receptors. Every zone has a combination of different receptors that are actually present in that zone. Because each odorant is detected by an ensemble of neurons that is widely dispersed across the epithelial sheet, some receptors detecting a particular odorant will remain functional when a part of epithelium is damaged by the respiratory infection. That is what we said. So then the sensory inputs in the olfactory bulb are arranged by receptor type, right? How does that happen? We will look at it very soon. The axons of olfactory sensory neurons project into ipsilateral olfactory bulb, whose rostral end lies just above the olfactory epithelium, right? These sensory axons terminate on the dendrites of olfactory bulb neurons within bundles of neuropil called glomeruli that are arrayed over the bulb surface. So this is the glomerulus. Glomerulus over here refers to an area where in the olfactory bulb wherein olfactory sensory neuron is relaying the information to the mitral or the tufted cells, right? In each glomerulus, the sensory axons make synaptic connections with three types of neurons: mitral and tufted relay neurons. These are the mitral and tufted relay neurons. The periglomerular interneurons, which encircle the sorry periglomerular interneurons and project axons to the olfactory cortex, right, which encircle the glomerulus over here. The axon of the olfactory sensory neuron terminates in only one glomerulus, right? What does that mean? That one sensory neuron is going to terminate in one glomerulus only. It is not going to divide and it is not going to go in different glomeruli. And similarly, the primary dendrite of each mitral and tufted relay neuron is confined to a single glomerulus, right? However, at the same time, in each glomerulus, in each of these, axons of several thousand sensory neurons coverage on the dendrites of approximately 40 to 50 relay neurons, right? What happens is that in this glomeruli, one sensory neuron is coming only over here, but there are multiple such single sensory neurons that are coming over here and forming a synapse with a single mitral cell, right? How then what happens that thousands of these sensory neurons will pass information only to maybe 40 or 50 of these mitral cells and tufted cells, right? So that is how the signal is getting relayed. From these thousands, now it is narrowing down to 40, 50 maybe. This convergence results in approximately 100-fold decrease in the number of neurons transmitting olfactory signal, right? We will study why that toning down is important. Right, this is a representation of axons of neurons with same odorant receptor converge on a few glomeruli only. Right, so if the odorant receptor was same, it is not going in all the glomeruli. It is converging on maybe one or two glomeruli only. And one odorant can activate many glomeruli that you can see over here. So how do these odor responses look in the olfactory bulb? Right, the organization of sensory information in olfactory bulb is dramatically different from that of the epithelium. Right, in epithelium, what used to happen? Different regions of the odor receptors are scattered across the epithelium, and different regions have multiple, sensory receptors that are present over there. Right. However, over here, what happens? The olfactory sensory neurons with same odorant receptor are randomly scattered in one epithelial zone. Their axons converge in few glomeruli at two spots, one on either side of the olfactory bulb. Right? So over here you can see this is olfactory bulb on one side, this is on the other side of the brain, right? And whatever information is being taken is taken, it like these glomeruli are bilaterally symmetrical, right? The number of glomeruli that are present on the right side of the brain are similar to number of glomeruli present on the left side of the brain also. And that information is from the similar kind of receptors are transmitted to the same regions of glomeruli across different regions in the brain also, right? Each glomerulus and each mitral and tufted relay neuron connected to it receives input from just one type of odorant receptor, right? So each glomerulus is receiving input from one type of odorant receptor only. The result is a precise arrangement of inputs from different odorant receptors which is similar between individuals, right? So this kind of an arrangement is similar between individuals. Because each odorant is recognized by a unique combination of receptors, each odorant also activates a particular combination of glomeruli in the olfactory bulb, right? So if these four glomeruli are being activated, that is because of a particular smell, right? Maybe if the smell was altered or odorant was altered, only these two will be activated, not these two, right? At the same time, just as an odorant receptor recognizes multiple odorants, a single glomerulus or given mitral or tufted cell is activated by more than one odorant also, right? That we have seen. This organization of sensory information in the olfactory bulb is likely to be advantageous in two respects. Right now, we will see how is this a particular kind of arrangement advantageous for us, right? So first is the fact that signals from thousands of sensory neurons with same odorant receptor always converge on the same few glomeruli and relay neurons in the olfactory bulb may optimize the detection of the odorants present at low concentrations, right? Uh, right, that was the first one. We will go through the second one also. In case of this, let us first go through what is periglomerular cell, what is this granule and mitral cells, right? So each glomerulus over here you can see encircled by periglomerular interneurons. Why are they known as interneurons? Because they are forming connections between two neurons, right? These periglomerular cells, they receive excitatory input from sensory axons and form inhibitory dendrodendritic synapses between mitral and tufted cells, cell dendrites that in the glomerulus and perhaps adjacent glomeruli, right? The periglomerular interneurons may therefore have a role in signal modulation because they are receiving excitatory information and they are also providing inhibitory signals to these synapses. They are kind of modulating this sense of olfaction, right? Then these granule cells are again, they are interneurons. They are connecting the or transmitting information between two neurons, right? And they are present deep in the deep in the bulb product provide and they provide negative feedback onto the mitral and the tufted cells. The granule cell interneurons are excited by the basal dendrites of the mitral and tufted cells and in turn inhibit those postsynaptic relay neurons and others with which they are connected, right? Then the lateral inhibition that is presented through this granule cell and the glomerular cell afforded by these connections is thought to dampen signals from glomeruli and relay neurons that respond only to odorant only weakly, thereby sharpening the contrast between the important and irrelevant sensory information before its transmission into the cortex. So this is another place due to the presence of these interneurons, both granule cells and periglomerular cells, the information is kind of being filtered, right? So they are, because they are providing some inhibitory responses. So if there was some very weak smell that was present in the background and a very strong smell which we should focus on, because odor receptors are coming for both odor receptors, so both are being activated. So it is filtering out the weaker smell so that we can focus on the more important or the smell that is present abundantly, right? This is the kind of system through which olfactory bulb is basically transmitting information to the olfactory cortex, right? So over here you can see that these mitral and tufted cells. They are going into the from the olfactory bulb and they are going to different brain regions, right? Including anterior olfactory nucleus, olfactory tubercle, piriform cortex, amygdala, entorhinal cortex also, right? And to different higher areas of the brain, right? Then you can see there is another organ over here that is the vomeronasal organ, right? It is a secondary nasal organ that is not present in us or in the higher organisms, but it is present in some invertebrates, for instance. It is an accessory organ, right? It is going into this accessory olfactory bulb, and the response is going into amygdala and hypothalamus, right? What this vomeronasal and accessory olfactory bulb does is it is responsible for pheromone perception in some of the animals, right? They can perceive these pheromones through their normal primary olfactory epithelium also, but also through vomeronasal organ, right? Then we come to taste, right? So we are done with the olfaction also or smell also, right? Now let's come to taste. As we know, taste have five submodalities or qualities, right? So we can taste basically five flavors is what, not flavors, we have five senses of taste, right? The primary function of gustatory system is nutritional. We will understand how. The human and other mammals can distinguish five different taste qualities. They are sweet or bitter or salty or sour or umami, right? Umami is a taste associated with amino acids. So the taste chemicals or tastins perceived as sweet are associated with food high in caloric value, while those sensed as umami are indicative of protein, right? So umami is actually a less common flavor that we can feel or we can understand, but it is indicative of protein, right? Consistent with the nutritional importance of carbohydrates and proteins, both sweet and umami tastes elicit pleasurable sensations in humans and are attractants for animals, right? So these are the two tastes that the animals or we will be attracted to, both the sweet taste and the umami taste, right? In contrast to that, bitter tastings, which are often found in poisonous plants, elicit an aversive response in an that is innate in animals as well as human infants and likely prevents the ingestion of toxic substances, right? So that is why, bitter tasting food, even food like coffee or tea, people develop that taste. Initially, if you give maybe black coffee to a person, they will probably not like it, or green tea to a person, they will with flavor. That is not true. However, taste refers strictly to five qualities encoded in the gustatory system, whereas flavor, with its rich and varied qualities, actually stems from a combination of inputs from the gustatory, olfactory, and somatic we have some olfactory response, then our taste receptors will have some response, then somatosensory, meaning the mechanoreceptors that are present on the tongue will feel the texture of the food, that will also have some response. So together this response will be summed up and it will elicit a flavor to that particular substance or food, right? But taste by itself is just the perception of the taste receptors that are present on the tongue, right? Not any other kinds of receptors. This taste detection occurs in the taste buds. In the gustatory system, sensory signals generated in mouth are relayed through the brainstem and thalamus to the gustatory cortex, right? So this is the brainstem, then it is going to thalamus, then to the gustatory cortex in the brain. Although the majority of taste buds in humans are located on the tongue itself, right? But there are some that can also be found on the palate. Palate is the upper, uh, this thing, the roof of the mouth, right? Pharynx, epiglottis, and upper third of the esophagus also. It might have some kind of a taste receptor, right? So these, you can see this is the tongue from which these taste receptors are taking responses via different nerves or via different pathways into this brainstem region, which is further taking it up to the thalamus and which is further taking it to the gustatory cortex, right? So these taste buds are clustered in the papillae on the tongue, right? So where in tongue are these taste buds present? It is present on special structures which are known as papillae, of which there are three types based on morphology and location. So there are three types of papillae that are present, right? So this is the tongue. These are the various taste receptors on the papillae on the tongue, right? These papillae, based on their morphology and based on where exactly they are present, they could either be circumvallate or foliate or fungiform, right? Each fungiform papilla consists of 1 to 5 taste buds, while both circumvallate and foliate papilla contain hundreds of taste buds. Right? So the taste bud is a garlic-shaped structure embedded in the epithelium. You can see this is the epithelium that is going. This garlic-shaped structure is actually a taste bud. A small opening at the epithelial surface over here is the taste core, which is the point of contact with the taste tips, right? Each taste bud contains approximately 100 taste receptor cells or taste cells. These are the taste cells, these ones. So each taste bud is containing around 100 of these cells, elongated cells that stretch from the taste core to the basal area of the bud. This is the basal area and this is the taste core. The taste bud also contains other elongated cells that are thought to serve as supporting function as well as number of the round cells at the base, which are thought to serve as stem cells, right? These are the basal stem cells. These cells are short-lived and appear to be continually replaced from stem cell population. So these T cells are not very long lived, right? They keep on forming and keep on dying. So that is why this population of basal stem cells is actually present, so that they can keep replacing these taste buds. The taste cells also resemble neurons in that they are electrically excitable, right? So although these taste cells are not neurons, right? In like photoreceptors, they were neurons, right? Over there in yeah, photoreceptors were neurons by themselves. These by themselves, these taste cells are not neurons, but they resemble neurons. Why? Because they are capable of being excited electrically. They also have voltage gated Na plus, K plus, and calcium or Ca2 plus channels and are capable of generating action potentials. Right? Then each of the tastes that we basically feel is detected by sensory transduction mechanism and distinct population of taste cells, right? Each of the 5 taste qualities involves a different sensory transduction mechanism in the microvilli of taste cells. We will see what they mean. There are, however, 2 general types, that is bittersweet and umami tastings interact with G protein coupled receptors, again GPCRs, whereas the salty and sour tastings appear to be involved with the specific ion channels, right? This is a representation of that. So you can see these are ion channels and these are transmembrane, 7 transmembrane protein structures, which are GPCRs, right? So salty and sour taste, they are dependent on this ion channels, and the bitter, sweet, and umami are dependent on different GPCRs. For bitter, it is the T2RS. For sweet, it is T1R2 and T1R3, and for umami, it is T1R1 and T1R3, right? So T1R3 is actually common between sweet and umami. Over here again, you can see a combination of T1R2 and T1R3 recognizing sweet tastings. Most of these are sugar names, right? Polysaccharide names or sugar names. So that is being recognized by these receptors, which recognize sweet tastings. Then the umami tastings, T1R1 and T1R3 recognizes umami tastings. Most of these you can see are actually all of these are some kinds of amino acids themselves, right? Then come the T2R receptors, which recognize bitter tastings, right? So these are some chemical names that are given and which are by nature they are bitter or they are a little toxic, and you can see that the T2R receptors responding or recognizing these particular tastes. The sensory neurons carry taste information from the taste bud into the brain. Each taste cell is innervated at its base by the peripheral branches of the axons of primary sensory neurons, right? Each sensory fiber branches many times and innervates the several taste cells within the numerous taste buds. the release of neurotransmitter from these taste cells onto the sensory fiber induces action potentials in the fibers and transmission of signals to the sensory cell body, right? These cell bodies of gustatory sensory neurons line the geniculate, petrosal, and nodose ganglia, right? So these are the three ganglia that are being talked about, in which lie the cell bodies of gustatory sensory neurons that have receptors on the tongue, right? The peripheral branches of gustatory sensory neurons in these three ganglia travel in cranial nerves 7, 9, and 10, right, as is represented over here. Okay. The central branches of axons of the gustatory sensory neurons enter the medulla where they terminate on the neurons. They enter over here in the medulla where they are terminating on neurons in the gustatory area of the nucleus of the solitary tract. Right? This is that region. In most mammals, the neurons in the nucleus transmit signals to parabrachial nucleus of the pons, right? Which in turn sends gustatory information to ventroposterior medial nucleus of the thalamus. So this is that region. In primates, however, these neurons transmit gustatory information directly to thalamus. So this is a representation of a human brain. So that is why direct information transfer is what is represented. From the thalamus, taste information goes to the gustatory cortex located along the border between anterior insula and the frontal operculum, right? The gustatory cortex is believed to mediate conscious perception and discrimination of the taste stimuli. The taste area of the thalamus also transmit information both directly and indirectly to the hypothalamus, a structure that controls feeding behavior as well as autonomic responses, right? So I have actually rushed through this portion, but we have talked about sense organs in detail in the previous lecture, if you remember, right? I hope you would guys have already watched that lecture before moving on to this one or skipping to this one. I just stopped sharing my screen, right? So this will be it from my end on in this particular course, that is basics of neurobiology. So the more molecular level of how neuron is functioning, how action potential is being generated, how is it being transmitted, how is memory consolidation happening, okay, and how is basic cognition happening. More about that we will study in another course that I will probably design to follow up this one. Right. So for now, this is it from my end. Thank you all for attending the course. Have a good day.